In flowering plants, pollination is an important mechanism that aids in transferring the pollen released from the anther to the stigma of the flower. If the pollen is of the right type, the stigma induces it to germinate and produce a pollen tube. The pollen tube, containing the two male gametes, then secretes hydrolytic enzymes which help the pollen tube to digest the tissues of the style as it travels down to enter the ovule through the micropyle, a minute opening in the ovule. Inside the ovule, the pollen tube encounters the embryo sac, the female gametophyte containing 3 antipetals, 2 synergids, an egg cell, and a central cell with 2 polar nuclei. The pollen tube now enters one of the synergids through the filiform apparatus, during which it ruptures and releases the male gametes into the synergid's cytoplasm. Of the 2 male gametes, one enters the egg cell and fuses with its nucleus. This fusion, known as syngamy or first fertilization, results in the formation of a diploid zygote. Meanwhile, the other male gamete moves towards the 2 polar nuclei and fertilizes them to produce a triploid primary endosperm nucleus, or PEN. Incidentally, this fusion is known as triple fusion, as it involves the fusion of 3 haploid nuclei. After the triple fusion, the central cell is called the primary endosperm cell. Did you know that syngamy and triple fusion together represent double fertilization? A phenomenon unique to flowering plants. Double fertilization is followed by several post-fertilization events which eventually transform the ovule into a seed and the ovary into a fruit. These events include degeneration of the antipetals, synergids, and the development of the zygote into an embryo and the primary endosperm cell into the endosperm by repeated mitotic divisions. Endospermic development is of 3 types.
Mitosis is a process of cell division that makes possible regeneration of body parts. To elaborate, mitosis is the separation of nuclear chromosomes into 2 identical daughter nuclei. It is divided into 4 key phases: prophase, metaphase, anaphase, and telophase. An additional phase called cytokinesis occurs after the complete nuclear division. Before a dividing cell enters mitosis, it undergoes a period of growth and rest. This period is called interphase. In interphase, the genetic material of a cell increases. Prophase. During prophase, the chromatin condenses and becomes short and thick to form chromosomes. Each chromosome has 2 sister chromatids joined together at the centromere. The nucleolus disappears and the nuclear membrane starts to break down. The centrosomes, each with a pair of centrioles, move apart to the opposite poles, forming a spindle between them. Metaphase. During metaphase, Microtubules of the spindle interact with chromosomes. This causes the chromosomes to move in a line along the middle of the cell or the equatorial plate. Sister chromatids are held at the centromere, confirming the presence of protein structures called kinetochore at the surface. The kinetochore holds the chromosomes and attaches them to the spindle.
Note-taking is the practice of condensing the reading material or passage in a brief note form, which is meaningful yet concise and facilitates recall.